This method is an interesting approach to study organo-mineral interactions because it fractionates soil organic matter according to its association with different minerals. It is a physical fractionation process, which means it doesn't alter the chemistry of the fractions. As a result, analysis of the fraction tells us something about the natural composition of organo-mineral complexes.
This method is really useful as we try to learn more about the ability of different soils to sequester carbon away from the atmosphere.
To separate the free light fraction, weigh 5 to 8 grams of air-dried sieved soil in a 50 milliliter conical centrifuge tube. Record the mass of soil with 4 significant figures. Using a graduated cylinder, add 35 to 40 milliliters of sodium polytungstate, or SPT, with a density of 1.62 grams per cubic centimeter. Centrifuge for 90 minutes at 2,500 times gravity in a swinging bucket centrifuge to afford a clear separation between the free light fraction and the pellet. Pour all floating and suspended material into a 250 milliliter polycarbonate centrifuge bottle. Ensure that the pellet remains firmly lodged at the bottom of the tube. Rinse the material adhering to the wall of the tube into the same polycarbonate bottle. Use a squirt bottle filled with deionized water, holding the tube almost upside down above the polycarbonate bottle. Set up a vacuum ultrafiltration line with 0.45 micron filters. Slightly moisten the filters and apply vacuum before tightening the funnels to avoid tears. Slowly pour the content of the polycarbonate bottle into the funnel of a vacuum filtration unit. Rinse out any